There we go. Cool. And some share into. Yeah, I think grab this. Get back from here. Okay. All right. Cool. Okay. Let's see. Also, it's backwards. That's all right. I don't think anybody's watching me. Oh. Thank you. All right. Thanks for the invitation and excited to be here because there's a lot of, I think, work that we can't do necessarily to validate a lot of the imaging that we're doing. That there's a lot of expertise here. So there's a lot of ground for collaboration to move a lot of this forward. So I hope some of this is interesting to you, and we'll see what we can do.
I think it's the other one.
The other one. We'll get there eventually today, I think.
No. Arrow keys, yeah, right here.
Yeah, we'll just go with the arrow keys for now. No.
Oh,
How about the mouse? There we go. Okay, we got something working. Okay, so the main focus of the first part is trying to image beta cell mass, and that's using a dopamine receptor ligand. And then I'll show a little bit about work that we're just starting to get off the ground, trying to do some antibody PET imaging. And that's related to T cells in the pancreas during either transplantation or at the beginning of type 1 diabetes. And then the back half, I'll sort of shift gears and we'll look at, if there's anyone interested in the liver, hopefully, we'll look at liver, adipose tissue, and brain imaging as well. So sort of, those are pre-diabetic, obese, looking at the cortisol stress activation pathway. And then I'll just mention a few sort of future projects that we're interested in. So I'm not sure if everybody's on the same page with PET imaging, but in our case, we have a radioligand that we tag some kind of drug that we're interested in. In this case, think of dopamine for our example. We have a radioactive drug that'll target the dopamine receptor. We inject that into the patient, put them in the scanner, and then you have this drug that, in this case, going to the liver. You'll then have a positron that will annihilate, and there's a little bit of a walk or a positron range away from where that actual target is, and that's important because that limits our image resolution in PET. You're limited to that positron range. It annihilates with an electron, and you get coincidence 180 detection on the camera, and we're able to sum those up over time in the scanner, and you eventually get, with reconstruction, a 3D image. So in this case, I'm just showing the most popular clinical scan, which is a glucose scan, looking at glucose metabolism. And you'll see, obviously, the brain, heart, and then a tumor there in the lymph node in the shoulder are all very active in glucose metabolism. But our interest at Yale is that PET is not just FDG. We have hundreds of compounds that we've injected actually into humans. So we have a large database of human scans that we can do. So we're interested in moving into a novel sort of PET tracer sphere. But in this first part, we want to know, can we develop a technique to measure beta cell mass in vivo? This would allow us to track different therapies for type 1 diabetes from early or pre-diagnosis, if we know they're going to get diabetes, and see if those therapies can prevent this. And primarily C-peptide measurements, as most of you know, are not necessarily a good measure of mass. They give us some kind of indication of what may still be going on in there. But we're really interested in tying that into a beta cell mass. So from a PET imaging perspective, we want to develop a radioligand target that expressed only in the beta cells. There's been a few targets before this that have been measured in some fashion or another that have also shown binding in polypeptide cells or exocrine tissue that doesn't give you a good reflection of just the beta cell mass. And importantly, that should be on both targeted into active and dormant beta cells. So there's going to be, well, at least we theorize that there's some residual binding in there that are binding to beta cells that are not necessarily active, but can give you that target for some kind of drug treatment. And I'll show some data to support that, hopefully. And you want a high affinity target. You don't want a drug binding to several different receptors, kind of a promiscuous target that's binding to a few different areas. You want it specific to that one receptor that you're hoping to measure. So part of the most important part of this from our perspective is, again, just to get everyone on the same page of what we're talking about here, is this: we have beta cell function, which is going to be in the red. Those are the active cells. And then we have dormant cells in the blue that are not going to be necessarily functional, secreting insulin, being supportive, but those increases and decreases in demand of even just the normal beta cell function can change over time in normal physiology. And it looks like you still have that same beta cell mass. So the functional measures, yes.
If you could measure, I mean, ideally knowing both is great, but if you could measure one versus the other.
I'll probably cheat and say that both is important, because I'll show some data later where we do measure both, and it gives you a characteristic of sort of different stratifications of patients and what they might benefit from. Because I mean, right now we're just measuring functional by C-peptide or proinsulin. That gives you a surrogate measure, but if you just do mass alone, then you may not know that you have some functional cells also. You're just saying there's a mass of cells there. You're not sure what they're doing. So I think it has to be some sort of combination. And obviously in diabetes, you're going to get this reduction in beta cell mass, and how much of that is active versus dormant is still up for debate and could be patient specific. So again, just a lot of times glucose tolerance test for beta cell function doesn't really distinguish between these two. So we hope that a PET measurement of just beta cell mass combined with a functional measure will give us a better picture of this. And if we're going to do this longitudinally, we can then track that in vivo and measure this increase in transplanted or pharmacologically treated beta cells. And just to show this on kind of this is ex vivo beta cell area that's been done after autopsy and showing C-peptide release. And I always like to highlight these three individuals with diabetes that show that there's three distinct masses here, but different functions. So the patient that had diabetes all the way on your right still had a very large beta cell area. So does that mean they have potential for more functional mass within that mass that they weren't utilizing? Whereas the person on the left, likely that was just their limit of how much functional capacity they have, since they're very low on the beta cell area side of things. And so a lot of this started for us because we have a lot of tracers improved in human use for neurological studies. And so we theorized that some of those tracers that are already approved for human use in neurons may also be useful in beta cells, since they share a lot of the same receptors and transporters. And other groups have published on the serotonin transporter, showing differences between healthy controls and type 1 diabetics. And I show this because there's a theme here of there still being residual binding in the type 1 diabetics in all of these tracers, some of which have been shown to have binding on other cells and some of which we're not sure yet, as we'll show in ours. But then the VMAT2 tracer as well from our group, same thing, they're showing this reduction in binding in the beta cells. So there's some sort of reduction in mass being measured by these tracers. In VMAT2, there was some off-target binding on polypeptide cells. Whether that is a stable thing across diabetics and healthy controls hasn't been fully explored yet. So that's something more in the validation step that we can look at. But we use this as preliminary evidence. There have been some trials looking at changes in VMAT2 in type 2 diabetes as well. But are there better specific markers that we know that are only bound to the beta cell? So we went back to a gene expression database in the beta cells, and we pulled out the top 5 genes that we know we have already approved human ligands. So we didn't want to start with rodent validation and move up. We can do these scans in humans already. So we have 2 different dopamine receptor genes, 2 and 3, 11 beta HSD1, which will end up coming back up in the second half of the talk, serotonin transporter, which is not surprising since serotonin precursor was already shown in humans, and then a synaptic vesicle protein A, which is a vesicle protein, likely on the vesicles that secrete insulin. And we have these 6 radioligands that were already approved. So we went and scanned one male, one female, and all of these tracers. And the dopamine, 3 dopamine ones were the most successful. But then if you look at the dopamine 3 receptor target, it has an affinity 50 times higher for the dopamine 3 receptor than 2. You can see that nice banana shape of the pancreas lighting up very hot. Whereas the one that has equal affinity for D2 and D3, you can't really see with the white arrows there. There's not as much uptake in the pancreas. So we think this is primarily driven by a D3 mechanism. And that bore itself out in the genetic data as well. In this database, at least, there was very high D3 expression in the primary beta cells, where there's none in the islets and the exocrine pancreas. We're still trying to validate some of this as well. But then we went back, and the role of dopamine in diabetes has actually been known for over 40 years. Just one example is if you infuse dopamine IV, you get these responses in glucose, insulin, and glucagon. Antipsychotic drugs have for a number of years caused diabetic symptoms by being antagonists of the dopamine receptor. Whether that's central or peripheral mainly is up for debate, but it eventually causes beta cell failure and downregulation of the insulin receptor, leading to some diabetic symptoms. And then bromocriptine as a D2 agonist has actually been used as a centrally acting anti-diabetic agent. So the role of dopamine centrally and peripherally has actually been studied for a while. So the idea of this is that the dopamine is actually synthesized in the beta cells itself, packaged with insulin, and then it's co-released with the insulin and acts as an autocrine signal on the beta cell itself to regulate insulin secretion. And the exact ratios of D3 to D2 receptors on the beta cell we're still not sure about, but I'll show some data on the next slide if I can get there. There we go. So this was just very preliminary staining. This isn't very complex multi-staining on the same slice. These are all different slices, but we showed co-localization with D3 and D2 with insulin. So there's definitely D2 and D3 on the beta cells. What the relative ratio is, we're unsure. But then there's no co-staining with D2 or D3 in any of the other endocrine cells. And then when we looked at a small sample of type 1 diabetic tissue that we had, there was no insulin and no D2 and D3 co-localization. So we think that this loss of insulin is also a loss of these dopamine receptors, at least in this small staining that we did. And then if we look at the comparison between type 1 and healthy controls, we see similar reduction in the type 1 diabetics that we saw in some of the other studies. And you can see nicely in the green arrows from the top row is 2 different axial and sagittal views of the pancreas. And you can see the reduction there. This is a representative image of actually the mean difference between the two visually there. But the interesting thing for us is there's sort of this shifting of the field in the last 20 years or so, that everyone said nobody in type 1 diabetes has any beta cells. But then with the Joslin cohort, there started to be some evidence of people living in their 70s and 80s that still had some residual beta cells at autopsy. And so that kind of fueled our active versus dormant beta cell theory. And we did arginine stimulus tests on all these folks. And you can see sort of 3 different representative patients here, where the bottom type 1 diabetic, no mass as measured by our PET imaging, and then no function on the stimulus test as well. Whereas the top in the green there, you see they have the highest, or that's the second highest, but second highest measurement in the PET but they have no function at all. So are there's just beta cells sitting there that we're measuring where they just have no function, whereas someone with a similar beta cell mass by PET has this functional release as well. So there's this sort of stratification of different patients, and a lot of them actually overlap with the healthy controls. So is this just normal physiology of them starting higher or lower, and then some destruction as well? That's still to be kind of determined. But the nice part about this would be, if that's true, then you have someone with no mass and no function who maybe could be a candidate for a transplantation. Whereas if that top person in the green, you could maybe stimulate those beta cells with some kind of pharmacological agent and restore the function if possible. And one of the nice things that sort of also corroborated this story was we went back and looked at pro-insulin and C-peptide. Yes.
Next slide.
Yeah.
And again, I'm sorry.
No, that's fine. Yeah.
So what's the age range on these subjects here, and are you confirming that they have actually type 1 diabetes, or what do you define that as in broader studies?
I'll show that we actually have the age distribution in a couple slides. I think it's 18 to 50 or something like that. We don't do any under 18 because of the radiation dose for imaging. I think I don't recall the exact criteria for type 1. I think we're just going by like the ADA definition of type 1 diabetes in that case. Yep. But the nice thing about this is if you look at the C-peptide and proinsulin, it's sort of this, they have the proinsulin distribution, and then the ability to kind of differentiate them with C-peptide is not there, which it was seen in a paper published by this group as well. If you look on the far right, you can see the C-peptide almost all on the 0 axis there, which mimics what we're seeing. We see a range of PET imaging, PET receptor binding, but no C-peptide. Whereas in the bottom middle green box, you can see that then there's a range of proinsulin in these same subjects, suggesting there's some kind of residual beta cell mass there that's not fully functional, which we see the same spread in our PET measurement and the proinsulin as well. And getting into sort of an age at onset discussion, because for a while it was considered that the duration of diabetes was more important to the beta cell mass, whereas it's now more thought that the younger the patient is when they first present symptoms, the worse off the drop off is going to be, the more precipitous that's going to be. And so this is a paper from a while ago, but it gives a nice indication of where at age of onset, 7 years old or less, already 68% of these subjects, 68% of them had beta cells in the islets. Whereas if they were diagnosed after 21, 100% of them had islets with beta cells. Whereas, and following them up 1 to 30 years later, only 6% still had those islets, where 71% of the older cohort still had functional islets. So there's definitely an age component to this, and that is shown here with our PET measurement as well. You can look at duration of diabetes, doesn't correlate great. I mean, you can see a downward trend, so obviously age is a factor there, but age of diagnosis is a much better correlation with our PET measurement. So we're definitely seeing those younger age of diagnosis is presenting with a lower binding in our PET measurement. So just to summarize, we think proinsulin and not C-peptide is more reflective of sort of those residual beta cell mass that could be there. These beta cells are not able to fully cleave the proinsulin to form C-peptide, and we think maybe our PET measurement can capture that. And age of diagnosis, we've kind of think is a strong predictor of our radioligand uptake as well. Whether this is an indirect indication of immune response or more severe destruction, there's lots of people, including everyone here, that is studying that. So we're moving on. We're doing this in a pilot study as well in type 2 diabetics. I'm really trying to distinguish now between D2 and D3 receptor sites, since some of those other D2 radioligands had high binding as well, but not as good as D3. There still may be some some interaction between these D2 and D3 receptor activity on the beta cells and maybe controlling beta cell mass in some manner. And we're trying to tie this in with hopefully a grant that should be coming this summer with TrialNet and getting people who are immediately diagnosed and scanning them a few months after diagnosis. And then hopefully in the longer term, we can follow them serially every year and see that kind of progression or hopefully not progression to diabetes. And right now we've started some islet proteomics where this is part of the grant as well. I'll just show you some of the proteomics on the next one, where we looked at different pathways. We haven't done the stressed islets yet. We'd like to do this and see how the islets get stressed and then see how these proteomic networks change. But if you look at dopamine receptor signaling in this healthy islet, you see all kinds of dopamine feedback related to the insulin receptor, dopamine degradation, catecholamine biosynthesis, insulin secretion pathway. So there's definitely some interplay here between the dopamine receptor and insulin system. And now we can hopefully look at quantitative proteomics to see how these proteins are changing as we stress them in different ways and try to mimic diabetes. We've had some trouble getting this off the ground, but we're trying to sort of mimic some of the imaging mass cytometry that's been done before. And I highlight in green the CD8 positive over there because I'll talk about something we're doing with that with PET imaging in a second. But the idea would be there's a lot of these panels that are mostly focused on immune cells that we would like to add in. The highlighted bolded ones here are all PET imaging techniques that we have that have been proposed for beta cell mass imaging. And can we now look at those in the co-localization with other immune cells and different phases in type 1 diabetes progression? So just to reiterate, our kind of end goal would be then to follow this progression of the black line where you're following beta cell mass and not the blue line where you're following beta cell function, where it can variably change during the progression, and sort of be able to catch people along that line and either treat in several manners. And just to show the image as well that we actually do get the brain. So you can see the striatum here where we get two different dopamine, or to each side of the striatum, where it would be very interesting to then go longitudinally and see if the brain and pancreas are affected in different manners during the progression of diabetes, and look at sort of that whole body regulation of dopamine. And so this is a brief sort of talk about the technical developments that are coming down the pipeline in the next couple of years, just because I need to introduce that to talk about T-cell imaging in the pancreas, because the number of T cells are going to be very low probably in the pancreas, and we need very high resolution and high sensitivity to be able to inject not overly high doses to measure this. So if you look at a conventional PET scanner, if you inject say 1 millicurie, you're only collecting the yellow lines. So you're getting a lot, like 50% of the actual counts that are coming out of say the brain in this case. So you're actually losing a lot of the signal and a lot of information there, where there's a total Explorer PET that's coming out now that, say you inject 1 millicurie, you have this long axial field of view, you're getting much more information for the same injection. So you're increasing your signal to noise 10, 40, or 50 times, depending on which organ you're imaging. And our lab at Yale is now going to be in this longer axial field of view brain scanner. So if you compare that to the conventional PET scanner, you're getting more information in just the same volume. And some of these other detector advances are more to just make the reconstructions more accurate. You can co-localize where that annihilation is actually coming from. So it really gives you very nice imaging. If you look at the top pancreas here, that's on the older scanner, that's probably 6-7 millimeter resolution. We just scanned our first couple subjects on the newer, this is 3 or 4 resolution. And the pancreas doesn't really excite you that much in this case, but if you look down here on the right, you can actually see the stomach wall lining and some of the dopamine receptors that are in the stomach and some of the gut there. So that's really kind of really nice advances there. And then this is the Explorer total body PET that I just showed you on the last one, where the far left there is a 20-minute scan, and then the far right over here is about 20 seconds. So that allows you to either scan very short and get the same image quality, or if, say, for a CD8 imaging technique that I'll show you next, you could scan for an hour and get much more data for the same injection, or even lower the injection and scan the same length. So there's some variable parameters that now we can play with. If we want to move into, say, a pediatric population and try to do this, we can lower the dose 10, 20-fold, and then try to image pediatric patients as they develop diabetes. And so some of that now is motivated by the teplizumab trial, where they showed delayed progression of type 1 diabetes. That's an anti-CD3 therapy. It also showed alterations in CD8 positive T cells, but a lot of these measures were peripheral blood measures of T cells, not necessarily the T cells in the pancreas directly. Whether that's reflective of the changes in the pancreas needs to be determined, but we want to look at those CD8 and CD3 cells directly in the pancreas. And we submitted a JDRF grant on this with collaborators from City of Hope. Anna Wu developed the CD8 antibody. I'll show you some of the clinical scans they've already done in cancer for that on the next slide. But the motivation of this was, there's a couple papers that show this co-localization of CD8 and CD3 in, I believe this is a NOD mouse. Oh, sorry, this is an allograft of the mouse kidney, where they've transplanted the allograft on the kidney, and there was this immune response into the kidney allograft of CD8 and CD3. And in another study, they actually showed an anti-CD3 therapy in a similar way. Teplizumab delayed the infiltration and destruction of this transplanted graft. So we want to try to be able, if we could see sort of this infiltrate in vivo, that would be very nice. And so this on the left there is a CD8 positive T cell PET scan. In this case, they're giving a cancer therapy, so they actually want the T cells to attack that cancer cell or cancer tumor, which you could see in the top right. You don't see a bright spot in the jaw yet, because that's pre-treatment, but the bright spot in the jaw that you see on the bottom right there, that's the immune infiltration into the cancer, hopefully to destruct that tumor that's present. And in the case of our proposal, we can label zirconium with CD8 and then indium with CD3, and you can actually co-inject these at the same time, and then scan one on PET and SPECT, and being able to use the energy windows of the photons you're detecting, you can actually separate these and create an overlay image to get that dual information at the same time. So I'm going to shift gears. That's sort of the pancreas focus. Yep. Question?
Can I just ask you a couple of basic questions?
Yeah, of course.
Not a radiologist or an authority in this, but can you explain to me the probes? And you know, I know there are different probes. There's a carbon-11, there's fluorine, there's gallium.
Yeah.
I assume you're talking about carbon-11.
Yeah. So for the PHNO is carbon-11. So typically when a radioligand is first developed, the carbon-11 is used because it's the easiest to replace on the structure. You're just replacing a carbon with another carbon. So that's typically where radiopharmacists will start with that. And then as you want to move to the clinic, you need the F18 typically, or a gallium that have a couple hour half-life. So you can actually ship it to the clinic. And like FDG, for example, they could ship it across the state lines, and there's many centers across the country that'll just ship it to hospitals and then they use it. And that takes a little bit more, there's a little more tricks to the chemistry to sort of force that fluorine molecule into the drug structure that you're trying. And that can have effects on your biodistribution and other things. So there's sort of whole fields trying to decide those techniques. But
And then what advantage does your probe imaging the beta cell have over what's been used? I know VMAT2,
yeah.
which is another.
So the VMAT2, there was some binding on the polypeptide cells that was not specific to beta cells. The the other one, serotonin precursor that I showed, there's actually binding on some of the other endocrine cells as well. So it's a good measure of islet cells, but not necessarily beta cells specifically. There's been a lot of good work with GLP-1 as well. And they're sort of in the same phase we are of showing the same validation steps, and it's all sort of coalescing around. I know they just published a paper showing the same kind of age and sort of residual binding measures that we were looking at as well. So we're all sort of coalescing around the same idea. It's just sort of making sure we're not measuring any off-target effects. Thanks. So I'll shift gears a little bit. This is my interest in looking at sort of the stress component of overall cortisol enzyme, the enzyme that activates cortisol in obesity. So this was actually the focus of my KL1 and a couple other pilot fundings, but we've developed a target, and I showed this before. We tested it in the pancreas, which didn't really have good uptake, but can we create a measure of 11-beta-HSD1 throughout the body that sort of gives you the activating enzyme measure? And you can look at that in different tissues. For us, we're interested in liver, adipose tissue, and brain. And so just a little bit of background for that. With chronic nutrition, you get liver accumulation in the liver, lipid accumulation, hepatic triglycerides, then these lipid-induced hepatic insulin resistance, fatty liver disease, obesity. And a lot of these pathways are exacerbated by cortisol. So cortisol stimulates gluconeogenesis in the liver, promotes further differentiation of adipocytes, and then regulates stress both long-term and acute in the brain by mediating kind of inflammation in the glial cells. So how these kind of all interplay is of interest. And part of the issue is that you say, why don't you just measure blood cortisol? It's sort of the same idea as the beta cell mass. We want to know the intracellular actions of cortisol. So there's intracellularly, this enzyme activates cortisol, and then it binds to the glucocorticoid or mineralocorticoid receptor, and then it's deactivated. So a lot of this happens within the cell itself. And so if you're measuring peripheral cortisol measures, you might not be measuring sort of what's going on in these different tissues. You're just getting kind of a system-wide approximation. And luckily for us, the enzyme is the same in humans and rodents, so we have a good way to validate this in rodent studies as well. And some of the interest from fatty liver disease is a lot of these drugs have shown reduction in fat in the liver compared to control subjects, but you can see there's some correlation in the blue line here of the fatter their liver is, the more they lose fat with this drug. But you can look at the spread of these blue triangles is all over the place. So there's obviously some stratification that may further improve the use of this drug if we can target the right population. And this drug is the 11-beta-HSD1 inhibitors have been looked at in type 2 diabetes and largely failed, probably because it's too late in the progression of type 2 diabetes to have an effect. There's already insulin resistance and some other effects that are downstream that are complicating the picture. So can we determine appropriate clinical population? And part of this is motivated by even some of the studies that have looked at these, trying to measure this enzyme. They're trying to deduce how much cortisol was activated and metabolized through different pathways in urinary and venous samples. But even some of these venous samples, you can see if you're measuring the liver, if you're trying to measure the hepatic vein cortisol, you're still going to get some mesentery cortisol from the gut itself that's being dumped into that pathway. So it's not a good way to discriminate between liver and adipose tissue in the visceral component. So can we directly measure that? And I think we now can. I'll show some data that we're showing that both in humans and rodents, we're binding to this enzyme specifically. You can see very high uptake in the liver, brain as well. The arm is just the administration of the tracer, nothing funky going on over there. But the drug that we're using here is actually an antagonist for that enzyme that we've radio labeled. This is actually an F18 compound, so we have a little bit longer half-life if we were to move this to the clinic. And you can see here, this is sort of the typical progression of developing a PET tracer. On the left, it's the C11 compound. It's the MR on the far left, baseline in the middle, and then we use a blocking drug, typically say a cold drug, which is the cold version of the hot radioactive drug. And we inject that before the scan. If we show that we're blocking all of the hot radioactive drug, we know that's specifically bound to the target we're hoping it's bound to. And then just once we move to an F18 component, you can see MRI on the top, test, retest. So we'll scan them like a week later and see if this is a stable measurement with no interventions. We want that to be a stable measurement. We want to know that we're measuring the same thing week to week. So they both demonstrate specific binding to that target. We looked at some of these binding just with the mRNA reference to the Allen Atlas, which is there's only like 6 subjects in there, but it did correlate well with the mRNA for that in different regions in the brain. And then this is just showing again that the brain measurements of this enzyme did not correlate with peripheral cortisol. So we're measuring something specific in the brain that is not related to like a blood measurement of cortisol. And then this was another study retrospectively, so it's not perfect, but we went back and looked at all the people we scanned and noticed that there was a decrease in binding as they increase adiposity. And you can see that in the striatum here, it gets lower in the axial view. And we saw a nice correlation of this as well. And group differences were significant between lean, overweight, and obese. So that was sort of the pilot driving motivation to then move into the liver and adipose tissue, since we saw these differences. Just general overall reduction in the brain, but you can see with even the smaller groups, we see these significances in putamen, thalamus, hypothalamus, which is of particular interest in obesity. We also noticed an age correlation as well, which is typically more prominent in aging studies. People have shown aged rats and in aging cortisol secretion sort of increases over the lifespan, causing cognitive dysfunction in older adults. So this may have something to do with this enzyme, or we're still not clear. Wouldn't call this a trend. There was one subject that kind of drove this increase in the older group, but like I was saying a second ago, this is sort of the idea that cortisol causes this accelerated aging. So there's a group of high cortisol rats versus low cortisol, and they're much more slower at escaping mazes and some other cognitive function tests as well. So we moved into a preclinical model. We're doing obese and lean Zucker rats, and for a lot of the PET imaging we take blood samples and try to use that to model the tracer going in and out of the tissue of interest. In this case, we used an image-derived blood sample because you can't take large blood samples from animals continuously throughout the scan. So this is just showing that we can measure the activity in the abdominal aorta and then use the measurements in the liver and adipose tissue. And we basically take a ratio of what's in the liver and adipose tissue and what's in the aorta. And that gives us some measure of what's specifically bound versus what's going in and out of the tissue in the blood pool as well. And similar to the brain, we give a blocking drug. We show we're actually blocking what we see in the liver. And in this case, this is brown fat in the green arrow here. Then we take that out, do Western blots. We've shown a good correlation of our liver protein. We're working on some other Western blots now to quantify that. And this is just showing again, we image them over time, take this quantification. The red line is basically the blood pool. And then the other ones are different tissue volumes that we're measuring how much activity is in there. And essentially we just take a sum of those activities from 40 to 60 minutes and then take a ratio. So that gives us a good idea of what's going on. And just showing that now we can see this enzyme going down in liver, brain, and brown adipose tissue as they increase in weight. I'll show in a second that that's the opposite of the humans in the liver, but it turns out that people have already studied this in this model, and they showed mRNA drops off in this specific Zucker model. That's probably not a good model for human obesity. They get genetically obese very fast and all sorts of other interactions. So we're going back and doing a different model now in the preclinical animals. So just cross-section of lean to overweight to obese individuals. And you can see a stepwise kind of green, blue, and an orange for overweight and obese. And we see a nice correlation of the higher this enzyme that is activating cortisol is in the liver, the higher the BMI and the higher the fat fraction in the liver, which we've actually done by taking the same area of interest that we're using for the PET quantification. And we just do a histogram of that, and then we can play around with that and look at how many of these voxels are fat. So you can see this subject over here, say has fatty liver, a lot of fibrosis and normal liver tissue. The rest could be blood pool, lymph nodes, other stuff that's within the lymphatic tissue. And we can use that then to say, similar to what we were trying to say in the beta cell mass, we can look at how much fat they have in the liver. If they have fat and a reasonably high uptake of this enzyme, can we give them an inhibitor to then try to reduce the fatty liver? Whereas someone on the far right here, they don't have a lot of fat in the liver and they have very high enzyme uptake. So if we're giving them the enzyme, it really might not help. They may be too far along in the progression of the disease at that point. So it could be a way to stratify different patients. So obesity, we see the downregulation of this enzyme. We think that that's a protective effect of the increases in the adipose tissue and the liver. And we're looking at these different combination of phenotypes. So you can think of kind of this grid where you say, okay, if they have high enzyme, but low liver fat, that's probably maybe obese without fatty liver disease. They may be a candidate for protective therapy to try to reduce this cortisol that would progress them to fatty liver disease. But if you have high liver fat and high enzyme uptake, that could be a responder for fatty liver disease. Whereas if someone has high liver fat but low enzyme, you're not going to have enough enzyme there to target to stop the progression of fatty liver disease. So we're hoping that this maybe can be a sort of stratification of patients, maybe to be more effective in treating fatty liver. And this is just sort of an overview. Still playing around with this for a grant proposal, so it's a little messy. But the idea is that this enzyme in the top there, we're seeing a reduction in the brain, but we're seeing an increase in the liver and adipose tissue. And that's thought to be an increase in the liver fat that's then dumped into a visceral compartment, which then feeds back through this hepatic, feeds back more glycerol, hepatic gluconeogenesis, and becomes sort of this feedback loop in the adipose tissue and liver that then is being sensed in the brain somehow, probably through the vagus nerve or something, or an unknown peripheral signal. Unlikely cortisol, because that doesn't seem to be, it must be some other mediating factor. But seeing this downregulation in the brain is interesting. And now can we associate that with the increase in brain inflammation that's usually seen with obesity? If this cortisol acutely is supposed to be there to tampen down the inflammation is gone, then that can lead to the progression of inflammation within the hypothalamus long term and kind of promote further dysregulation and obesity. So just one slide summary of dopamine receptors in diabetes. Oh, I messed up the second one. That should be CD3 and CD8 in diabetes, for looking at different applications of transplantation and progression of newly diagnosed, and then our interest with cortisol activation and obesity as well. And then just obviously a large group goes into this. There's a lot of staff that helps with both animal and human research and collaborators in endocrinology as well. So, thank you. Yeah.
The cortisol story is there. Have you taken circadian
So
things into account for that?
yeah, right now we just scan the same time, like 11 o'clock every day, to avoid that. I think in the early, before they paid attention to that in the first study, I think they looked at the basically the PET measurement didn't vary a lot with circadian rhythm.
I don't know.
But just to avoid that, yeah.
Cortisol does, but I don't know about the ends.
Yeah, and we're still trying to figure out how sort of stable that enzyme is. Right now, we're doing some studies in animals where we're trying to. We have an ASO knockdown of the enzyme, so we can kind of see liver only. And does then that affect fat and brain within a week, within 2 weeks? Trying to see if it's more acute regulation or if it's kind of steady and moves in one direction or another. Yeah.
There's an advantage to treating at a certain time.
Yeah, exactly. No, definitely. Yeah.
Jason, I would say that, there's this group called Teddy, which is looking at biomarkers of progressors in type
Yeah.
1. And I know Kendra, who Des knows well, has been looking at cortisol levels as a biomarker of progressors and non-progressors.
Oh, okay.
I really urge you to
Yeah.
reach out to her and see what data she has on hand for cortisol.
Yeah, that'd be great.
Helpful.
Yeah.
The other thing is, one of the areas where we've been interested in, I'm curious about whether you've thought about it, is the genetics of some of this, and whether you have any DNA or on your subjects where you've done some of the imaging.
So right now we don't have that, but well, we have blood samples to do just like regular blood measurements of glucose, insulin, and all that, but we haven't taken any. We tried to propose to do some like subcutaneous punctures of adipose tissue to then look at sort of DNA and other stuff like that, but haven't gotten that approved yet.
It's certainly an area where I'm interested.
Yeah.
It's
Yeah.
tough, right? Numbers wise.
Yeah, exactly.
But at least, when you think about the rate of disease progression, those really young subjects,
Yeah,
they're going to be enriched in higher risk HLA groups.
yeah,
And at least knowing some of that polygenic risk scoring, I think would be helpful.
yeah. I think it would probably help stratify some of our PET signal as well, even further. I mean, this is a small sample size, 9 and 7 for the first study, but I think talking to a few others today as well, trying to stratify even further in larger groups down the line would be beneficial.
Any other questions for Jason?
Say it in the chat.
Yeah, is there any?
Oh, I didn't check the chat. Yeah. Oh. Nope.
All right. More questions? Thank you, Jason.
Thank you.